Hello, my friends. Thank you so much for joining me. So, this is the third part of my biotechnology and biological medicinal products presentation. And in this presentation, it will be about quality by design. I will be focusing mainly on the conceptual and the concepts of quality by design. I will be applying it to the monoclonal antibody example, the fragmented monoclonal antibody example, but I will be talking mainly about concepts in this presentation. So as I said, the presentation is going to cover some of the concepts of quality by design. I'll give a brief introduction. I'll give some terminology descriptions and definitions. And then I'm going to talk about quality by design. I'm going to talk about the design of the molecule and the quality attribute assessments, which is important with regards to understanding and gaining an understanding of the drug product. I'm going to talk about manufacturing process development, upstream and downstream manufacture, the drug product itself, some control strategies, and then I'm going to talk about this case study of the AMAB. So this is the monoclonal antibody example where we have a quality by design approach to product development. So, introduction. So, medicinal products that are monoclonal antibodies are effectively, a form of biological medicinal products or biotech medicinal products. And they are more complex in their structures compared to chemical entities. Biological molecular medicinal products such as monoclonal antibodies are typically larger, really much more significantly larger than chemical entities, which are small in comparison. And they are typically heterogeneous. If we're going to compare them side by side, if you like, with chemical entities, you can get a highly pure chemical entities that's like 99.9% purity in assays in terms of your actual active substance. And the purity, when I talk about 99.9%, I mean it's uniform. There's no heterogeneity. It's all, a uniform state, and any minor changes in the structure, we consider it to be impurities, whether it be process related or product related impurities. This is completely different to biologicals, molecular medicinal products, which are larger, as I said, typically heterogeneous, and therefore they present the potential for more structural interactions. Yeah, because they're larger, there's more of a surface area, and we have to consider the safety and therapeutic nature and aspects, which are going to be more for biologicals such as monoclonal antibodies. So as a consequence of this, more information must be provided and evaluated during regulatory application. So maybe for clinical trials and maybe for marketing authorization applications, you're going to be providing a lot more information. The components that are used to manufacture biological medicines are themselves heterogeneous, and sometimes they are biological components themselves. Sometimes they're complex in their nature themselves. For example, cell substrates that are used as, if you like, the factories that manufacture monoclonal antibodies are quite complex. Do you know what I mean? And therefore, there is the potential for, this increased complexity that's going to produce more heterogeneous products, but there's also the potential for the introduction of contaminants, whether they be adventitious viruses and microbes or other cellular contaminants or you know, the contamination or the impurities that as a result of using cells to manufacture proteins. So there's a lot to consider and account for, and therefore the regulatory documentation for these molecules is significantly more. What have I got here? Therefore the subject matter is going to be larger. That's what I say, the regulatory documentation is going to be larger, more, and you have to consider a lot more. You have to consider the functional, structural, and biological perspective, because it's a larger molecule and there's more potential interaction. There's more of a safety considerations, as well as the efficacy considerations. So as I said, this presentation is an overview of the quality by design approach for monoclonal antibodies, and I will talk about the regulatory considerations, but I don't think I'm going to get enough time to do that, and so I'm going to probably talk more in depth about the regulatory considerations and what should be put in your regulatory applications in another presentation. So the crux of this presentation is going to be on quality by design. And basically, as I've got here, quality by design is an approach, and it's described in the ICH guidelines in the pharmaceutical development, and it's a systematic science and risk-based approach to pharmaceutical development to ensure that the product is high quality because of your deep understanding of the product and process. Quality by design it means that the quality of your medicinal drug product is built into the manufacture of your product, and you obtain that quality because you have a thorough understanding of your manufacturing process and of your drug product itself. So your objective is to have a high quality finished medicinal product, finished product. And you are going to build that into your manufacturing process so that the reliance on testing the products after you've manufactured them is limited to just confirmatory, or you may even remove them altogether and replace it with real-time release testing. So quality by design is applicable to monoclonal antibodies, as it is also applicable to chemical entities, and it's also applicable to cell and gene therapy medicinal products. And what it does is it provides assurance that the medicinal product is fit for purpose, i.e. it is designed for its intended use. So as I've got there, it does not rely solely on final product testing. It focuses on designing quality into the product from the beginning. So I put together a little pictorial representation of what it is about, and it's about having a good product understanding, a thorough product understanding, thorough understanding of your process. And ultimately, what you want is a safe and efficacious medicinal product. And you do this by having or putting together in your dossier, based upon data, a design space. And that design space will include your normal operating range and your control space. And that will be within your design space. So quality by design, the approach is about having a good understanding of your product, having a very good understanding of your process, defining your design space based upon data, based upon predictive analysis, based upon modeling. And that design space will include your normal operating range, but ultimately all of this has to be linked to the safety and the efficacy in the patient, whether it be in the patient in the clinic or in the patient on the market. So the next thing I want to just quickly run through, and I hope not to spend too much time on this, is the terminology, because we'll be using these terminologies. So critical quality attributes. So this is a physical, chemical, or biological or microbiological property or characteristic, and it has to be within an appropriate limit, range, or distribution, and it is there to assure, to provide confidence, if you like, on the desired product quality. And what is quality itself? It's the suitability of either a drug substance or a drug product for its intended use. And it usually includes terms or attributes such as identity, strength, and purity. So these terminologies were just taken from the ICH guidelines. So what is risk? So risk is a product of severity times probability, multiplied by well, it's a product of severity and probability. So your severity is the consequences and your probability is the likelihood of something happening. So in the context of a medicinal product and quality by design, it's looking at what might go wrong. So this is the attribute that is under consideration. So your product, the particular attribute of your product under consideration, what could go wrong? So it's the risk or the hazard identification. And what are the consequences? This is the severity. You know, how severe is it? Is it going to lead to, you know, all the cells in your cell substrate being non-viable? Is it going to lead to a product that is out of specification? What are the consequences of this particular risk? And what is the likelihood that it will go wrong? And that's the probability. So your risk assessment, I think I gave another presentation on this, on the quality risk management, but your risk assessment is identifying and evaluating the risks associated with your critical quality attributes, and you need to determine which factors have a significant impact on product performance and safety. And you can use risk assessment tools when you're doing that, such as failure mode effect analysis and your fishbone analysis and fault tree analysis. If you look at the ICH, I think it's Q8, I can't remember which one it is, quality risk management ICH guidelines, they provide a number of tools that can be used in performing a risk assessment and risk analysis, and also risk identification. So, critical process parameters, we just get through all the terminology. So a process parameter is some kind of a variable or factor. So, for example, the temperature of your air dryer or bed dryer, your buffer pH, your paddle speed. This might be a process of parameter that occurs during your manufacturing process. Now, if the parameter's variability has an impact on your critical quality attribute, which has already been predefined and pre-specified, it should be monitored, it should be controlled to ensure that the process produces the desired drug product or the drug substance. But we'll just stick to drug product for the time being, but it is applicable to drug substance. So you need to monitor it and control it if there is if the variability, if that parameter's variability has an impact in order to achieve your desired drug product quality. And as I said, this can be determined initially by performing some risk assessments where you will do a risk analysis and you will do a calculation against the predefined criteria, a risk evaluation, and then you will take appropriate actions to control or mitigate that risk by you know, monitoring or by controlling and limiting, say for example, the variability of that particular process. And there are various levels of process parameters in terms of their criticality on the attributes of your drug product. So it could be a key parameter that you're not really too concerned about variability, but it could be a critical process parameter in which that parameter variability of that parameter will lead to an impact on your drug product quality. So I just want to talk about design space. So when we talk about design space, what we mean is the multidimensional range of input variables and process parameters that ensures drug product quality. What does that mean? So traditionally, when we think about the impact of, say, for example, your drug product, the output, and a variable, say, for example, temperature, and the process, what is the process that may, for example, impact? We think about a single variable approach, yeah? And that's a very simple, a simplistic way of looking at it. Well, this is how I see it anyway. I'm pretty certain it's not like this in the ICH guidelines. But multidimensional is looking at a number of variables and process parameters to ensure your product quality. So your design space, you will use a multivariable approach with multivariate relationships, and you will have statistics and models in order to justify and put forward your proposed rationale for your design space. Yeah, so changes within that design space are not considered to be regulatory variations, provided they have, your design space has already been accepted during your assessment, during the evaluation of your marketing authorization. Usually that's where it happens. Provided it's already been accepted by the authorities that this is the design space, and you will have your normal operating ranges. If you have excursions outside of your normal operating range within your design space, they're not considered to be, so for example, an out of specification or a deviation. You know, you don't have to submit, say, for example, I think in the UK it may be a batch specific variation, say. You don't have to justify it, because you just have to monitor it, because your design space has already been validated and approved by the regulatory authorities. So it gives you that kind of flexibility, regulatory flexibility and operational flexibility when you're manufacturing your drug product. Right, so as I said, quality by design is a systematic understanding of, your product and your process, your product qualities and your process, your manufacturing process. And you need to evaluate and understand and refine your formulation and your manufacturing process. So what this involves or includes is identifying through knowledge, prior knowledge, through experimentation and risk assessments, the material attributes and the process parameters that can, have an effect on your critical quality attributes. So you really need to determine the functional relationships between your materials and your process. And once you've understood that functional relationship, you can then do a criticality assessment in order to determine what are the critical quality attributes that you need to measure. And ultimately, this will lead to, or is what's driving this, or this is extrapolated to the safety and efficacy of the drug in the patient, whether it be in the clinic or in the marketplace. So using this sort of enhanced product and process understanding, and you can use it in combination with quality risk management, you can then, and that is to perform risk assessments, yeah, so you perform risk assessments, and you can then establish the appropriate control strategy, and I'll go through that in the next slide. And this will then lead you on to, understanding and putting forward, proposing a design space, and then within that design space you can even have real-time testing, real-time release testing. So here's that pictorial, if you like, representation again. So your prior knowledge and that can be platform technology, that can be historical data, that can be literature studies and literature documents. Yeah. Your experimentation, so that can include clinical studies, animal studies, in vitro studies. Yeah. And these will enable you to be able to perform a risk assessment. Yeah. And through that risk assessment, you can then perform your criticality assessment. So your criticality assessment is where you want to understand what process parameters, what material attributes will impact on your product's safety and efficacy, i.e. the product's quality, the ability of the product to perform its intended use. And you can then rank the criticality, what is high in terms of criticality and what is low. And whatever is high in terms of criticality, that's what you need to control. Yeah, and you need to establish a control strategy to do so. And that control strategy, as I said in the previous slide, it could include your design space, where you can then, and it can also include your real-time release testing. And it can include it can and will include a whole load of other elements, but this is overall what I mean by quality by design, the quality by design approach, where you are looking at your design space. So you're looking to get a design space based upon an understanding of your material attributes and your process parameters. And this is it starts off as maybe, for example, platform technology, maybe scientific literature, etc., and clinical, animal, and in vitro studies. But with time, you build on that knowledge and you improve and provide more data to support your design space. I just want to, I think I'm still going on about terminologies, but I do need to talk about control strategy. So your overall control strategy, yeah, is designed to ensure that your manufacturing process consistently delivers the product that meets your established, if you like, criteria or specifications. So your established quality ranges and it ultimately assures drug safety and efficacy. So that relationship between your attributes, your drug product attributes, and safety and efficacy, that relationship, whether it be direct or indirect, is something that would already have been established. Yeah. So what have I got? A planned set of controls derived from current product and process understanding. Yeah. That provides assurance on process performance and product quality. So let's just go through that list. So the elements can be material attributes. So your raw materials, where you source your raw materials are important. You need to consider the lifetime sourcing of your raw materials if they are really important for your drug product. You want to ensure that you can, with regards to continuity of supplies, yeah, you need to ensure that there are procedures in place with regards to your suppliers, particularly if they contain, your raw materials contain human or animal derived products, yeah. You need to have the company or your suppliers need to have risk mitigation procedures in place. You may need to take risk mitigation actions yourself, such as, for example, considering using synthetic materials, raw materials, if it has no impact on qualities. You need to ensure that your third-party agreements are in place with your suppliers, so that they have to, I mean, these are standard practices, you know, that they have to ensure that they advise you if there are changes, if they make any changes to their manufacturing process. Yeah, so that, you know, you can, you know, that could potentially impact on your qualities and safety of your product. So Not all of this information that I'm saying here is going to be in the regulatory dossier, absolutely not. But you will have pharmaceutical quality systems in place, and the documentation will be there. And this is good documentation practice, you know, to ensure that these documents are available to be inspected by health authorities when they provide certification for your manufacturing site. Yeah, so any changes in your raw material specifications, say for example any variability in your raw materials that may impact on your final quality, on the final quality of your drug product, you need to understand and you need to control for, and that will be part of your control strategy. So you need to understand the impact of variability in the specification of your raw materials and starting materials on the final drug product itself. Yeah. And this is what control strategy is all about. So we talk about your raw materials, starting materials, intermediates, and drug substance. All of these things have to be controlled for. You have to understand the variability of the specifications on your drug product. So the grade of your starting material or your raw material, or the quality of your starting material or raw material is important. You know, your specifications and acceptance criteria from your sourcing suppliers have to be stated. You may require, you know, you might have to have, and usually there is, certificates of analysis. Your suppliers need to assure that they're not providing you with potentially contaminated with adventitious agents, as well as you need to do your tests as well with regards to that. But you know, there needs to be agreements in place to ensure that you know, the quality of your drug product, you mitigate any potential risks. These are the things that we talk about when we refer to control strategy. So when I was talking about criticality assessment and looking at the functional relationships between materials and the intended use, this is kind of the sort of things that you would need to be looking at. The next thing on the list is process parameters. So your process parameters is, you know, you need to sort of understand the functional relationship between a process step, say for example, and your product. And this is again part of your criticality assessment, where you're determining what steps can impact on your product quality, and you need to be constantly doing this. And this because ultimately it is related to the quality of your drug product, and ultimately related to the safety and efficacy in the patient, whether it be in the clinic or the marketplace. So you need to understand what the key process parameters are and what the critical process parameters are. And the critical process parameters are what you need to be monitoring, obviously, because they're going to be critical and they will affect your critical quality attributes. They impact, there's a direct or maybe even indirect relationship to your critical quality attributes. So the critical process parameters are what you need to monitor and control, because variability in those parameters may impact on the quality of your final drug product. Next thing on that list is the specification. So your product specification is really for release. Yeah, it's for release, and your release, well, the release is confirmatory. It's batch release data or batch analysis data is after the product has been manufactured. So it's really confirmation of the fact of the quality of your product. Stability specifications, again, is part of your control strategy. You know, sometimes you may need to perform changes in your manufacturing process, and you would put batches on stability, those manufactured batches where you have changed, you know, say for example, change something in your manufacturing process, and then you would compare a comparative stability analysis, where you compare the stability data generated on the new batches compared to the stability data generated on the previous manufacturing process, to show that over time there's been no change in the long-term stability of your drug product. Yeah, because sometimes you can make changes to your manufacturing process, and all of your testing are within specification, and it appears that there's been no impact on the quality of your drug product, but it's only when you place it on stability that you can see that, oh, okay, there are some changes, and perhaps we should or we should not implement this change. So stability is an important part. Stability testing and stability specifications is an important part of your control strategy. So for product specification, the sort of tests that you put in there, you have to meet the regulatory requirements. The health authorities will have minimum testing that should be included in your specification, and these will include things like identity, purity, potency, or your assay, and microbial testing such as sterility, bioburden, mycoplasma, and looking at your viral clearance or your virus. And you also need to include in there your product specific tests. So these are tests that specifically relate to your product. So the inclusion or the absence of certain tests must be justified, as must the acceptance criteria or the limits or the distribution range applied to each test. All of these things have to be justified based upon data. Characterization and comparability. I kind of talked a little bit about comparability. Comparability is where you make changes and you want to show that those changes don't impact on the quality, safety, efficacy. And you start off with the approach at looking at quality and looking at experimental, laboratory experimental data, but you may find yourself having to justify and make and do additional I don't know, experimentation, which could include animal studies, or it could even include additional clinical trials. So comparability testing is a subject all by itself, I think, but nevertheless, this is part of your control strategy, and it's something that you need to take into consideration. Characterization, again, is important. You need to be able to characterize your product, and you need to be able to characterize your manufacturing process. Characterization of your manufacturing process includes looking at every unit operation and looking to ensure that you have performed your viral characterization, looking at the viruses that have been part of your, I don't know, your manufacturing process, or viruses that could be adventitious, relevant viruses, and just looking at your clearance strategy and your inactivation strategy, and producing the relevant documentation to support that in your regulatory application. Facilities and equipment. So this means using clean rooms and sterile facilities, ensuring the steps are taken to reduce cross-contamination or the introduction of adventitious agents, as I've previously said. There is an EU guideline, GMP guideline. I think it was updated last year, and this relates to the manufacture of sterile medicinal products. If you're going to manufacture within the European Union, and if you're going to use supply to human beings in the European Union, you're using sterile medicinal products, which will be the case for monoclonal antibodies, then you need to comply with this guideline. So your control strategy ultimately ensures that your manufacturing process can consistently deliver a drug product that meets the established quality ranges and ensures drug safety and efficacy. Yeah. I think I'll just quickly go through this. And what's the purpose of having a control strategy? Well, as I've just said, it's to ensure that you consistently meet the desired quality, safety, efficacy, and a well-defined control strategy allows a manufacturer to have a design space. You have that design space. It means that you have more regulatory flexibility, and also you can implement continuous improvement strategies, because you are continually adding data to that design space based upon process performance data, based upon data on your drug product. So you continually improve and can make changes to your manufacturing process that result in improvements in qualities, and not just remaining stagnant. So let's look at specific control strategies that we need to consider for monoclonal antibodies. So this includes some, but it's not an exclusive list. So the sort of things that you need to look for is things like glycosylation. You need to look for aggregation. You need to look at impurities such as host cell protein impurity. So your host cell protein in the example that I've given in a previous presentation was Chinese hamster ovary, you know, and looking to see if there's, you know, any impurities from there, and looking at your viruses and your viral clearance studies. So these are the sort of things that you need to be looking at in order to control for the quality for monoclonal antibodies. How do we get continuous improvement? You know, so I mean, I have alluded to this in previous slides, but you know, what is the process? And this is the accumulation of data. So process performance qualification is continually monitoring and gathering data to build a complete understanding of your manufacturing process. And you can do this through data. Well, once you've obtained that data, you can analyze that data. You can use predictive strategies to justify your design space, to expand your design space, and to produce, if you like, more controls on your design space. You know, so you can do this through collecting data, and the data that you collect will be through your process performance studies and verification studies and qualification studies, through your annual product reviews, which is a pharmaceutical quality systems requirement, and through also looking at post-approval change management. So post-approval change management is where all of the changes, in this case maybe that are within your well, maybe not within your design spaces, they're planned in advance, and you obtain prior approval from the health authorities before you can implement those changes. So you have a number of changes, you put it in a protocol, you get that protocol approved, and then you can implement the changes when it, you know, in a way that's optimal, sorry, for the manufacturer. And this is where you have regulatory flexibility and your pharmaceutical quality systems in place, where you have your change management system, so that any changes that are made are thoroughly assessed internally by the company before subjecting those changes to regulatory evaluation if it's required, or before implementing. So all of these support continuous improvement and is part of this control strategy, quality by design approach. Right, so there's some things I need to talk about. When you are developing any product, I think, but specifically biotech and biological medicinal products such as monoclonal antibodies, you start off somewhere, and you start off with your target product profile. Now, this is a living document, and really what you want to do is you want to try to obtain as much information as possible on the drug product that you're designing. So you're going to design a drug product, and you have to have a strategy for the design of that drug product. You need to be to understand what the long-term goals are from a therapeutic perspective, and this is the purpose of your target product profile as well, and other documents are then derived from that target product profile. So this is a living document. It is continuously updated as we get more information. It has a multidisciplinary approach to it, so that you have disciplines, or you have inputs from various departments, such as marketing department, such as you know, the clinical department, you know, the health technologies department. So basically it provides a roadmap for all of the work that you plan to do on your product. As I say, you start with with the end in mind, but you you have a plan. It's almost like I said, it's a roadmap. It's a strategic document. It was widely developed by industry, and it's useful for patient groups, health authorities, and marketing teams. And I believe it was initially developed by the FDA, but there is some information from the World Health Authority. And again, the target product profile, it includes the clinical aspects, it includes the drug product aspects, and it can include your information, like, for example, stability, in use data, how to administer the drug, transportation and compatibility, etc. And the reason why you have this target product profile is because you want to, as I say, use it as a strategic document that can be used to assess, where there is an unmet need, the therapeutic potential of a drug, i.e. the ability to create something that is new, some kind of new intervention or a treatment or new technology. And the overall goal is to improve on the standard of care in the member state or in the country that you're, or region that you're looking to introduce this new technology and this new intervention into. But you also consider things like, for example, reimbursement, and how what's the probability or the possibility of that people are prepared, or companies or countries are prepared to pay for these medicinal products that could be very, very expensive in the short term, but in the long term very valuable. And how is that value going to be assessed? So this target product profile is a key document that is used to develop and used to think about the end in mind, as well as right at the beginning. Yeah. So, as I said, you have to think about or have a good, thorough understanding of your drug product, and you have to design your molecule. We talk about it as a molecule, but it's an antibody, if you like. It's this monoclonal antibody, but you have to design it with that long-term objective, but you also have to understand the properties and the characteristics, if you like, of your molecule. And this information is gathered in another living document, which is derived from your target product profile, and this is the quality target product profile document. And again, as I said, it's a living document. It's updated as more experience and expertise is gained about the product and the process. And you really should be considering it as a tool that enables you to design a high quality drug product right from the conception throughout the life cycle and the development of the drug product. So throughout the clinical trials and throughout the marketed medicinal product. So so your quality target product profile is a prospective summary of the quality characteristics of a drug product. And it will basically have all the attributes that are under consideration. So it will have your proposed quality acceptance criteria and some of the methods that you're using to measure them. So your molecular design, so these are your design features, and you can get that information, or you may start off from a platform knowledge. Yeah, you will also perform risk assessments. These are right at the initial stages. Yeah, and as you gain more experience and gather more data, your risk assessment will be based upon better data analysis, but you have risk assessments on your quality attributes, and then you have to select the quality attributes that are critical and provide a rationale for the selection of those critical quality attributes. And these are the kind of things that you need to consider for monoclonal antibodies. So they will be aggregation, glycosylation, deamidation, oxidation, etc., etc., whole cell proteins, DNA. as listed there. So this is the sort of thing that you will have on your quality target product profile, which is derived from your target product profile, but specific to the quality and the design of your product, your monoclonal antibody product, which are specific, or which have and hold specific information that is relevant for you producing, and for you having the right product knowledge, and from that knowledge being able to characterize and develop an appropriate manufacturing process. So your manufacturing process development is the next thing that you have to consider, because that's the whole purpose of quality by design. It is about your understanding your product and also understanding your process. So your process understanding is important. Process description and characterization, again, is important. And you need to provide this documentation in your regulatory submissions. Your manufacturing process is really sort of, I don't know, arbitrarily, I guess, divided into two sections: your upstream manufacture and your downstream manufacture. And I tend to think of your upstream manufacturers where you're manufacturing your molecule, or your your product itself, in this case the antibody, the monoclonal antibody. Whereas your downstream, I tend to think of it, and it tends to be in my experience, but obviously, I don't know everything, and there's a lot of other approaches out there, but it tends to be about purification. It tends to be about concentration, removal of contaminants, viral inactivation and viral removal, and then your final drug filling your final drug product in its final presentation. So your upstream process characterization, I've listed all of the things that we need to consider here. So it will be your seed expansion, your bioreactor, and your harvesting of your protein product. You need to define your design space, and again, you need to have a control strategy for the upstream manufacturing process, and this is going to be product specific. Your downstream manufacturing process, you will use other kinds of chromatography, not just the cation and anion exchange chromatography for purification, but you will use other resin derived chromatography for for purifying your product. Viral inactivation studies, so you need to be looking at potential adventitious virus contaminants. So just because you haven't found any viruses doesn't mean that you don't have to test for it. You need to do a risk assessment and look at all of your potential materials that could contain human or animal materials or raw materials, and what potential viruses would be in there. And you need to be able to qualify, what's the word I'm looking for, characterize your manufacturing process to be able to either remove or inactivate those viruses. The ICH guidelines have, they have sort of, I don't know, what's the word I'm looking for, they've categorized various types of viruses, whether these viruses be relevant viruses or whether they be model viruses. And they've also provided guidance with regards to what to do when you're performing your viral clearance and viral inactivation studies. You need to be able to, it's really in-depth sort of study, and it's the sort of thing that is very important, obviously, because you don't want to contaminate your drug product. You don't want a contaminated drug product, a viral contaminated drug product. And yeah, there are various strategies for viral removal, such as filtration methods, but you do have to have documented in your dossier your control strategy for your downstream manufacture. So I've just talked about the differences between, in this particular slide, the differences between your quality by design approach, specifically for upstream process, because that tends to be where the differences are. I mean, it's well established the sort of techniques that are used in your downstream purification concentration methods, but the difference is in the approach. So for quality by design, you have a thorough process understanding based on prior knowledge and product specific experience. Now they talk in the traditional upstream process, the process understanding is limited to a product specific empirical information, i.e. what we've obtained in the real world, what we've obtained from the data that we've generated on the product. Whereas quality by design, you may introduce platform knowledge, you may introduce other experimental data compared to the traditional approach. I don't know whether this traditional approach is always is sort of used. I think that this is like old school in my opinion, but it's there. That's what it states. I'll tell you where I got this document from, this table from in a later slide. So for quality by design, you have predictive relationships for process parameters and quality attributes, and you use statistically designed experiments, yeah, and you then propose your acceptable operating conditions, and these are expressed in terms of the design space. Whereas the traditional approach, you may do some experiments conducted on single variables approaches, and you might have, say for example, overlook other parameters, interactions. Again, I think that is a very simplistic way of looking at the traditional upstream approach, but personally, that's based upon my own experience. And then your rationale approach for establishing your control strategy is supported by your thorough process and product understanding, yeah? And and in the case of the traditional approach, your control strategy is based on your prior experience and precedent. That should be. So your product quality is controlled primarily by end product testing. I don't think that's the case, certainly for chemical entities, but I'll just give you where I got this from, and then you can make your own mind up. So here are some more information. I think you can look at this in your own time. So basically, this approach, the quality by design, is a continuous improvement approach. It involves the use of multivariate approaches for process verification. It is a much more complex and all-encompassing approach that allows for this design space to be based upon predictive data analysis, based upon real data, based upon platform knowledge, and based upon continuously accumulating data and continuous improvement. Again, I don't want to repeat what I've already said, but I think the most important point that I wanted to stress here is that the importance of developing or determining that functional relationship that links your material attributes to your process parameters, and that's what oh, I think there's a mistake there. That is linked to your critical quality attributes. Yeah, so really it's about determining what that functional relationship is. Right, so I want to talk about AMAP, and AMAP is a case study. So this is a comprehensive document that was created to, give the industry and I think health authorities a structured approach with regards to developing and manufacturing of monoclonal antibodies, and particularly in the context of quality by design. And it was developed by the CMC Biotech Working Group under Bioforum Operations Group. So this was first published in 2009. So the goal was to provide a hypothetical practical example of how quality by design principles can be applied, and it was in response to the regulatory expectations that were outlined in ICH Q8, Q9 and Q10. So the pharmaceutical development, the quality risk management and the pharmaceuticals quality systems. So it was in response to those particular you know, expectations from competent and health authorities. So this is it. And this is where, you know, the upstream quality by design approaches, I got this from this particular document. So this is the document, and it's a case study, and it's a really useful document, but it is a large document and it should be used as a reference. I think there's about 278 pages in this document. So it's a systematic risk-based development of monoclonal antibodies, yeah. And as I said, it's basically to align, at the time anyway, to align industry practice with the regulatory expectations. And it just illustrates how prior knowledge and risk assessment, because there's a lot of examples of risk assessments, how it can be used for process understanding. And it also provides a model for regulatory submissions that incorporate all of the quality by design approaches. So this is just an extract taken from this book as to what their objectives are. And it's listed there: demonstrates the implementation of principles of quality by design, leverages the significant knowledge base from both commercial and investigative monoclonal antibodies, shows the application to drug product and drug substance, provides illustrative examples, demonstrates the science and risk-based approach, and shows that there are many ways to implement quality by design. And, you know, why is it relevant? Well, for biopharmaceutical companies, it serves as a reference model, especially if you're developing monoclonal antibodies and biosimilars. For regulatory affairs professionals, it helps us to understand and integrate quality by design principles into our regulatory submissions and our regulatory applications. For researchers and academia, particularly, it provides a practical example of process and design and risk assessments for biologics in industry, in manufacturing, and industry training and education. Yeah, it's widely used in industry training programs on bioprocessing and regulatory compliance. So this is where you'll get it. This is the website, ISPE. So this is the International Society for Pharmaceutical Engineering. The FDA and the EMA, I think, also have technical reports, which I haven't included in this presentation, but you may want to look at them there. Well, thank you so much. I think I kind of sped through some of this because I'm conscious of the time. I will do another presentation on the regulatory sections, I think, that need to be updated with regards and what information should be placed in those sections. I think I might do another presentation on that. But thank you so much for your time. I hope you enjoyed this presentation. And if you have any questions, please don't hesitate to put them in the comment section. Thank you and have a nice day. Bye-bye.